Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley, and Cervantes Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy, and Cervantes.
Hey there, it's Proteomics in Proximity in 2023 for everyone. So today we're very excited to talk about a set of samples that were part of several clinical trials. So it's a pharma publication with Eli Lilly and Boehringer Ingelheim. Very exciting results where they dug into mechanistic biology after a successful clinical trial and, frankly, a blockbuster drug that has opportunities for repurposing. So let's go.
And those of you that are watching on video may be noticing that Cindy's got something in her lap and something that really you cannot avoid, which is a golden retriever puppy. So
5 weeks old.
Cindy, how in the world do you have a 5-week-old golden retriever puppy in your lap? I mean, how does that work?
So through much encouragement of you and Cervantes, I have actually brought this little gem on camera. Never nice. Perform with puppies or children, right? But this little guy is about 5 weeks old, and so we're actually raising them to go to wonderful families, and they are, just a genetics project to improve the breed. So I'm very excited about it.
Applied genetics, and you cannot see a golden retriever puppy without just thinking, oh my gosh, do they get any cuter than that? They really don't. They really don't. Look at that little guy.
And for those of you who are on audio only, maybe you'll go to the YouTube channel and check this little gem out. He's adorable. He's just waiting for his next treat because he is a golden retriever after all.
Yes, driven by treats. On the dog, on the sort of canine genetics piece, then you're actually, as a trained geneticist, Cindy, then you actually are quite sensitive, right, to the different weaknesses of the different sort of genetics of golden retriever in particular, right?
Yeah, and the dramatic opportunity that innovation in genetic tools has provided for keeping the breeding population as broad as we can, right? Because we talk about bottlenecks, and bottlenecks we measure them in an effective population size measurement. We try to estimate, how many real full chromosomes of single individuals are there in the population, and being able to test for some of these breed-specific variants that are causing challenges within specific breeds, like golden retrievers. Being able to measure those and know who's a carrier, who's not a carrier, and keep the animals that are carriers in the breeding population. Just make sure we don't breed them with other carriers, right? So it's good to have that diversity in there. And we used to just not know, right? So that's kind of how I ended up in this, was from my time at Illumina and my excitement about any opportunity to just make a small impact to improve something like this little creature.
It sounds like quite an interesting hobby, right? That you can connect, your interest in dogs and a particular dog breed along with the genetics of it, and then being able to do something about it, such as, bring along a 5-week-old puppy onto a podcast.
My husband says I don't do anything in second gear. I'm full throttle every time.
Speaking of full throttle, so getting back to the paper, the title, right, is, the drug in question is something called Jardiance, and the generic name is a mouthful, empagliflozin. And the title of the paper is Effective Empagliflozin on Circulating Proteomics in Heart Failure. Mechanistic Insights from the Emperor Program. First author, Faiz Zanad. Last author, Milton Packer. And I understand that Milton Packer is both at Baylor and Imperial College of London. So with that intro, and this was published in what journal, Cindy? Is it Circulation?
It's European Heart Journal.
European Heart Journal. Thank you.
In December 2022. Yeah.
So it was only published in December. And Cindy, you want to go ahead and introduce it a little bit further?
Yeah, sure. So as I mentioned, the clinical trial was not including Olink. This was post-clinical trial, and we've seen that several times in pharma. We've seen it more than we can talk about, but several times in publications. And so one of the notable publication examples of this previous to this empagliflozin, Jardiance, a little easier to say, was an Alnylam paper in hereditary amyloidosis. So that paper, again, was a post-successful clinical trial where the researchers were very keen to understand and unlock mechanistic biology. And I think that's what we're going to talk about today, some of the exciting understandings of some of these inhibitors and how they're implicating downstream effects, how other pathways are implicated in these downstream effects, which are unveiling some possibilities in other disease areas, which I think should be exciting for anyone.
And the term Jardiance may be familiar to a lot of us, right? In that we may know somebody who's on this particular drug because it's a blockbuster, right? It's some 4 billion euros in sales in 2021 from Boehringer Ingelheim and Eli Lilly. And apparently approved in 2014 for type 2 diabetes, and then in 2016 for type 2 diabetes and cardiovascular disease. And so what's really interesting, I think, from the point of the title of the paper was mechanistic insights, right? So maybe, Sarantis, maybe you can talk a little bit about that. Why mechanistic insights?
Because remember, Sarantis does affairs of the heart. Yes, Sarantis.
Thank you for reminding me this.
I just love that.
I think they test more than 1,000 samples.
Yes.
First of all, with our Olink Explorer, that includes more than 1,500 proteins. And the nice thing is, again, going non-invasive, looking in the plasma of a patient and be able to understand mechanistic insights. I think that was really exciting for everybody else here. It's much easier than dealing with biopsies. And it's more also painful and less invasive, less painful and less invasive, obviously. I think the goal here is to understand how this inhibitor that's specific against the SGLT2 protein. The SGLT2 protein is a co-transporter of glucose, and this is for this makes it important in type 2 diabetes. And they wanted to see what is the effect of this inhibitor at the heart failure, how contributes to reduce fibrosis, for example, in heart, and this reduce heart failure.
So this particular inhibitor, SGLT2, you're saying it transports glucose, but then somehow it helps your heart, right?
Yeah, that's the thing, because I think that this transporter protein actually is through the glucose is influencing energy, probably related things, or stress related, oxidative stress related pathways, most likely, or autophagic pathways. And at the end, after the analysis that they have done, I think they conclude that there are 30 roughly proteins that they are differentially expressed already by the 12 weeks of the treatment.
Now, Cindy, you showing the puppy in the full screen like that, I mean, that's really distracting. But I'll let you distract me.
I don't think the audience can see it if I'm not talking. But now that I'm talking, I mean, look at that face. So I was just going to say, this, as you mentioned, Sarantis, the autophagic pathways, right? What strikes me is, and they talk about this in the discussion, they talk about the impact on cardiomyocytes as well as the renal tubular cells. And what they say about future study just strikes me, as well as the autophagy, right? It strikes me as so similar to what like David Sinclair and Walter Longo talk about in terms of pathways in longevity, right?
Yes,
the sirtuin pathway, the mTOR pathway, right? Fascinating.
Interesting.
Actually, this is what fascinates me, because it's involved the sirtuin. That sirtuin is like a heat stack. It's a histon deacetylase that reduce the expression of the genes, and is involved in autophagy because regulates the expression of autophagic genes. And
Now,
most of the proteins a lot of proteins, they are regulated by the sirtuin complex, actually, by the sirtuin activity in this concept. That's really exciting for me, being epigeneticist.
But to hold for that thought for just a minute, we're referring to autophagy.
Yes.
Wait, Dale, we can't go on from this because I have to say that we have sirtuin-1 in our expansion to our 3K. And they talk here about future studies, right? So I'm super excited about that. The mTOR tour as well. So we've got some of these, named proteins that need further investigation in our expansion. And of course, we're going to be expanding further in 2023. And now you can talk more generally. I just had to make that plug.
And it's really interesting, this connection with longevity, right? This with healthy aging. And here we're talking about, two really common diseases, type 2 diabetes and cardiovascular disease. And you just say, and it's all connected, right? It's all a system.
Well, it's the concomitant inflammation, right? In both of those diseases that are really, I think, tying some of this together. And again, we're on a podcast. We can speculate all we want.
Yes.
And we love to do that. So please, we're just loving this.
But on that term, autophagy, how would you define that? What is autophagy?
I mean, it's if I can say, it's a Greek word.
There you go.
I love that. That's a great way to start.
I'm proud of this. It's autophagous, means that I eat myself in a way, in a simplified way. And this is actually what happens. It's a normal condition that is happening. Is under the harmony of the cell, because there's a constant reproduction and autophagic situations happening in order to keep the equilibrium, again coming from the Greek philosophy and Aristotle. And I think what is happening is we create an autophagosome where the proteins segregate. We have the autophagosome, and then close to the autophagosome is going the lysosomes, and they decompose in amino acids, for example, that they can be reusable. This is a turnover.
In other words, you're talking about trash collection.
Yes.
Right?
Well, go ahead, Dale. Sorry.
Damaged proteins and damaged cellular components
exactly.
are in these autophagic vesicles we're talking about. And you're saying they're next to the lysosomal compartment, and lysosomes are just grinding up whatever's there. Exactly. And so we have a selective autophagic process, which is frankly collecting trash and getting rid of it. But what happens, right, is what we're saying is that this particular SGLT2 inhibitor affects autophagy.
And it mimics nutrient reduction, right? Which mimics fasting. And in much of the fasting research, there is, certainly in mice, it's well documented. In people, it's just from imaging, I believe. But again, back to these researchers in longevity like Walter Longo, but that the organs are seen to actually kind of contract slightly during the fasting phase. And the theory, I think, is that these trash collection events are happening to move out cells. And if we're, again, speculation, if we're thinking about cancer cells as these growing cells that aren't dying on the normal schedule they should, and some of that fasting research suggests that fasting during some cancer treatments can be helpful in cancer treatments, that this is even more exciting in my sense about longevity. So.
Absolutely.
But I think Dale was talking to you, Cervantes.
No, I agree. It's great. And the only thing I will need to add is is close connected to energy production. For this, mitochondria plays a really important role.
Yeah.
And of course, in longevity and aging-related diseases and aging-related studies, mitochondria is one of the focus of the researchers, right?
So let's stop right there, right? So we were talking about SGL2 and autophagy.
Yes.
And so the autophagy, meaning there were particular proteins in that Explorer 1.5K panel that were associated with that, and they saw changes, right, in the circulation because the autophagic mechanism was affected by the drug. Do I understand that correctly? It was like what 1, 2, 3, 4, like 5 or 6 proteins?
IGF BP1, right? TGM2. I mean, these are ones that are also implicated in several other 2022 studies that are just corroborating exactly what's being seen here. Out of Iceland, out of Germany.
So just on that autophagic mechanism, right? We're talking about 6 proteins that were affected in the circulation that were measured by Explorer 1536. And what you're saying is that even though we're talking about heart health, we're talking about mechanism inside that cardiomyocytes, right? The heart muscle, it's actually reflected in the plasma.
Exactly. That's great. That's a great point, Dave.
Because then I'm like, well, you're talking about the organ. What's happening inside these myocytes, the trash collection? But you're saying that because of the drug, because of the sort of effect on these particular 6 genes, we can actually make a conclusion. This is how it works. Do I understand that correctly?
That's perfect.
And then you have other genes now you're talking about before you launch into the energetic side of things, because you're talking about mitochondria now.
Yeah, I mean, genes that are connected with, as I mentioned, with reflux, with let's say autophagic reflux. With autophagic flux are connected to mitochondrial
autophagic
function.
Flux. Flux meaning sort of the motion of the trash collection machinery. Yeah,
and this is like what is happening. And this is actually connected to mitochondrial, but also there are other genes connected to oxidative stress, inflammation, that they're also affected, and we can see the proteins that they are dysregulated.
So you're talking about these other genes that they've listed in one of their figures, TGM2 and IGF-BP4, apparently involved in what? Cardiac renewal, repair, and energy metabolism. So is this whole related to the mitochondria just because we know the function of these genes?
I think at the end, yes, but I can speculate. I mean, but I'm not so sure how is the mechanism of action of these genes particularly, but for sure at the point they will connect it to, since there's oxidative stress and since it's related to stress, for sure they will be connected to factors like stress factors that they are dysregulate functions like mitochondrial functions and other nuclear or cytoplasmic regulations, actually.
And again, it's fascinating from an energy usage point of view, right? To be thinking in terms of mitochondrial function. I mean, it's not just something that you hear about every day, and yet more and more attention is being paid to mitochondrial function, and the flow of energy through systems. It was at ASHG years ago. There was a plenary speaker from UPenn. I'll never forget it because he's talking about energy and cancer in a genetics meeting. But it was I never really thought about those connections and how important it is. And here it is right in the center of this figure is exactly that in terms of energy use, repair mechanism, metabolism, like you mentioned, oxidative stress, which is a huge topic, right?
Yeah, hypoxia factor, for example, activation of hypoxia factor that can upregulate the series of cascades, right, leading to apoptosis, for example, or something. It's a really fascinating and really
Can you
complicated.
Actually define apoptosis too? That's a good term to. Did we already define that? That's a great one.
I think they didn't. I mean, they don't see apoptosis, of course.
No. I just mean you mentioned it. Let's define it.
Okay. This is the apoptosis is not the programmed death, right? At the end, it's not the programmed death. At the end, what leads to apoptosis? This is, to my knowledge, this is something that is not programmed death at the end, and it's
I think apoptosis itself is the self-regulation of the natural death of cells, or am I flipping that?
Sure.
Technically, it's considered programmed cell death. Programmed cell death. And that Cindy's correct in that with particular triggers, a cell basically goes to terminal differentiation of death, right? Yeah. And it's a particular mechanism.
Thanks, immune system.
Yeah, so it's a particular mechanism, right?
This is what some of the tumors are able to, or one of the mechanisms that tumors are able to evade, right? Some of the tumors that should be apoptosing, or whatever the term of the adjective term is, that they just stick around and reproduce and then start taking away blood supply and Yeah. Well, it's interesting, right? We're using terms like autophagy and apoptosis, and these are all Greek
words.
There we go. Yeah.
So there we go. We have our resident expert here. And I mean, resulting from this particular kind of mechanistic understanding, what could you say is the so what? I mean, now that we understand that these sodium glucose cotransporter inhibitors, right, are able to improve cardiac health and able to improve renal health via these particular mechanisms, what conclusions then do the authors make in terms of the so what?
Yeah, I mean, they see the repositioning of the drug, right? That is the conclusion at the end. What they can see that this drug that used to be for actually diabetes 2, now can have an effect on heart, and they know why this is happening, right? And for sure there will be follow-up actions, target other molecules and other pathways, but this is a nice indication to see why there's an effect on the heart.
And they conclude more proteins. And there's a few, hypotheses generated in here, and there's some that I think are well corroborated by this study, which is exciting. But these are, I can't even imagine how much money went into these clinical trials, and to see extra value come of these samples is, I love that.
Yeah, and in particular, Cindy, this Emperor Reduced trial had 3,730 patients enrolled. Emperor's Preserved had 5,988 patients enrolled. Yeah. And from each, they chose about 600 from each to have a total of about 1,160 or so samples to be analyzed via Olink.
That reminds me of another point that I was that I'd written in the margins as I was reading this. And that was, they're bringing together these two studies that probably weren't collected with the plans of doing Olink proteomics in mind. This was 2015, 2016, right?
Yep.
And yet they bring these together with each of their concomitant, that's my favorite word today, apparently, with each of their pre-analytical differences, right? Maybe they were collected and spun down at different times. And yet, even with the more restrictive false positive rate in this study, about 1/5 of what it is, but being even more stringent, the signal just screams out. And I think this is a great lesson for those of us who are geneticists who worry about the variable nature of proteins, that some of these biological signals are so remarkable that even with that pre-analytical variation, it's a beautiful, beautiful story.
And to think that, these signals are in blood, right?
Yeah,
and it
accessible.
Could be accessible.
Good point.
And then you say, how much money and effort was, expended? As a rough number, we had almost 10,000 patient samples, right? From the two original clinical trials. Use a rough number like $50,000 per patient. You do the math, 50,000 times 10,000. All right. That's $500 million on a clinical trial. You just say, wow, Dale, how do you get that number? Well, 50,000 times 10,000. And then you say, okay, well, the additional value of the conclusion of this paper is to look at mechanism of action, right, for cardiovascular health. Then the question is, well, could we just apply this particular drug only for cardiac health, given, right, that they know how this works? And one of the sentences from the conclusion is, proteomics is rapidly emerging as an innovative approach to gaining potential insights in the mechanisms of disease and drug action.
My favorite sentence right there.
I mean, it really is, the why, why study proteins now? I mean, that's a really good question. Why study the circulating proteins now is, all the work that went into developing an inhibitor to this SGLT2, right? And then to not really understand a lot of how it works, other than, well, it reduces heart failure in diabetes patients. It's like, wow.
Yeah, it makes me think about so many of our neuropsychiatric drugs and how little we understand about their mechanism of action, but how desperately we needed them for years, right? And so I have a lot of optimism about being able to use these same tools. And we're, always on the lookout for CSF paired with plasma cohorts for our customers who are, especially a lot of pharma, who are very keen to understand these mechanisms. I mean, remember Eli Lilly's, like the last blockbuster drug like this that I think of was probably Prozac, right? I mean, probably they've had some in between that I just haven't been paying attention to, but man, what a big market that still is today, right? And how important is it for us to understand sort of how these things are working?
Yeah, and
so
for
again,
us to live through that.
I take it off the main point here, but hopefully it's interesting to you guys as well.
Well, I think many in our audience are familiar with somebody who's on Jardiance, or they've heard of it. Yeah. And you mentioned Prozac. We lived through, books were written about Prozac, right?
Yeah.
And so it changes society, right?
Yeah, it was. Wasn't it on Time Magazine as as Man of the Year or something? I don't know. It's like in our water supply for a while. Yeah, we got fish on Prozac, right?
Yeah. Well, interesting paper, great discussion. Cindy, thank you for sharing your puppy with us.
That was great. It was a great conversation.
Look at that.
Till next time. Take care, huh?
Take care.
Bye.
All right.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.